All right, I think we can start now. Good morning, everyone. Welcome to our first webinar from the Research University Group, Biosensor and Chemical Sensor, UKM. My name is Izati. I'll be your moderator for today. First, without further ado, I would like to introduce our esteemed speakers. It's my pleasure today to introduce Professor Chang Chia-Ching. His current position is a professor at the Department of Biological Science and Technology from National Yang Ming Chiao Tung University, Taiwan. He received his Bachelor of Science in Physics from National Tsinghua University, Taiwan, and proceed with his PhD in Life Sciences in the same university. He went to Johns Hopkins University for his pre-doctoral fellow and later to Academia Sinica for his postdoctoral fellow. So his research interest varies from conducting DNA development, characterization and application, bio-nanotechnology application, and new functional bio-nanomaterial development. Okay. So among his honors and award is in 2022, he received a Veblo Fellow from the USA. And also he has become and elected as a fellow from International Association of Advanced Materials Sweden, where he received the 2022 International Association of Advanced Materials Scientist Award from Sweden. He also received a Future Tech Award from the Ministry of Science and Technology Taiwan in 2021. And he received National Innovation Award for Research Innovation and Clinical Innovation. And he has received an Outstanding Fellow Honor Award from his university from 2021, 2015, 2011, 2020, and 2012. He is also a research fellow in the Institute of Physics, Academia Sinica. So his CV is very impressive now, okay? Without further ado, I would like to invite Prof. Chang to deliver his presentation for today entitled Nano-thin Film Electrode Development for Infectious Disease Detection, Typing and Anti-Infective Drug Screening. Prof. Chang, the floor is yours.
Good morning, ladies and gentlemen. Thanks, Izati's introduction. And today I will briefly introduce how we developing the nano-thin film electrode for biosensing. And then we use this biosensing electrode to detection the infectious disease. And we also found that some of the disease may mutate rapidly, so the typing will be important. At the same time, we also found some nature pairs, such like the virus and their receptors. And by using this compare, and then we can screen some drug or lead, which may affect those virus infection. So that's the overall about what my talk. Before go to the detail, first of all, I would like to introduce about my lab. My lab is the Bio-Nanotechnology and the Biological Physics Laboratory. And we are dealing from DNA molecule and the nanoparticle and to the real animal experiments to study the some photodynamic therapy and in animals. Or we also use some nanocarrier to efficiently deliver the CRISPR system and to see whether we can build the disease model and for in the future. Okay, and first of all, in my lab, and we are developing some novel functional material, including the nanodiamond. And why we use the nanodiamond? Because the diamond is pure carbon material, and their size is small. And in my lab, and we will focus in the diamond, which size below 6 nanometer. And then they can be a good carrier, and they can bond either DNA or proteins, and which can be uptake by the cell freely, and then they can use as the drug delivery, gene delivery, or some which can recognize, or if we label with the growth hormone, and they can recognize the cancer cell surface. And the most interesting part is that some of these kind nanodiamond, and which can be may containing some nitrosol inside their structure. So they can be induced by the laser irradiation and make their conformational change from the diamond to the graphite and induce the nanoblast. And this kind of nanoblast can make like the effect like a nanosurgery and kill make a tiny pit on the cell surface and then make the cell apoptosis. Okay. And if this material conjugate with the DNA, and then such like CRISPR system in our system, and we found that our transfection efficiency is about 10 times better than the current existing CRISPR system. And another topic in my lab, and we can refold the largest protein, such like the lipoprotein B. And this is a 500 kilodaltons protein, and it's the only protein in the LDL. So we can refold them and reconstitute their structure. During the process, we can integrate some drug or some material, nanomaterial, such like the iron oxide, and then which can be used, the nano iron oxide, which can be used for the MRI image contrast reagent. If we use this one and to deliver some anti-cancer drug, may enhance their efficacy, or reduce their side effect. And at the same time, we also assemble and make some nano drug or hydrophobic drug, such like the curcumin, and self-assembly and to form the nanoparticle. And this kind of nanoparticle and become relatively hydrophilic. And by combining with the photosynthesizer and the frat pair, frat linker, this one can also not only for the cancer drug and also containing the photodynamic therapy effect. So this one is one of our topic. And okay, so back to the topic today. I think every people know that we are suffering about the COVID-19, and although people may think that the COVID may not cause big outbreak or not. However, currently they are still remaining and affect a lot of people's alive. So during that time and when it's outbreak, even it's COVID-19. In fact, its outbreak is around the 2020. During that time, and the people think that how can we detect this new disease and how to find them? So it will be interesting to it not only interesting but also the people think it's important to detect that. So how people doing in general for biosensor, and in general it's containing the three different components. First of all is the samples. Okay, the samples are, for example, the COVID, the virus is the sample. However, they may from the spill or from other discharge, or that will be that is what we can detect, and or saliva. And these samples need proper receptor, and these sample and the receptor is kind of a pair of relator. So it's important, especially for new disease or novel virus. It will be important to find their binding pair. So the most common one is that if it's possible, and the people can can use the antibody to recognize those viral particle or bacterial specifically. But this one may take time. And some others, such like the drug, and can be detected and or can interact with the DNA, and we will discuss later. And in this, when those molecules interact with each other, and it's important to let our detection system to transfer the signal. So there is a transducer, and the people may use the FET, field effective transistor devices, or nanowire devices, or nanoparticle, or some optical or electrical, and such like the electrochemical devices. And so when those signals can be converted to the optical signal or electronic signal, and by using the amplifier and the converters, especially the ADDA converter, currently their resolution is high enough, and our signal amplifier, their efficiency is high, so we can detect their signal easily. So by using this signal processor, and the electronic system can detect such signal. So, but the most important part, right now, this kind of electronic system is mature, and with a lot can be available for a lot of commercial company. Yeah, but the problem is that how to find the pair of relator, and how to make it a good probe, means the transducer part will be most important. Okay, so based on the literature search, there are electrochemical based transducer, or optical based transducer, or mass based transducer. And by comparing with these different types transducer, and we found out the electrochemical sensor is relatively convenient and rapid, and do not need very expensive instrument. In my lab, and we decided to use the electrochemicals based to developing this electrochemical biosensor. Okay. For the biosensor, and the electrode or the probe will be important. And in general, such similar to the blood sugar test chips, or the gold electrode, and the people may think that they need a short detection time, and they are stable, or their sensitivity is high, and their detection limit is low, and their cost is low, and they can simply operate, and of course portable. So, but the key step is that we have to find some functional biomolecule, and then immobilize on it. So it becomes a bioconjugation problem between the biomolecule and the electrodes interface. So in a conventional way, and in fact, our lab, and we have studied for over 10 years, and this is the typical electrochemical electrode, and gold electrode, and which you can find in a lot of different company. And here is an example. And this one, for instance, and we purchased one electrode, and those gold electrode diameter is about 2 millimeter, and then you can find out their price is around $250 US. But before we use that, the student need to polish the surface to remove the non-related material on it, and then we still need to activate their surface, use the acid agent, And use after isolation, and the people try to generate a lot of 111 structure on the gold electrode. Why? Because these kind 111 structured surface and may have more functional side, and which can easily to conjugate to make the reaction to bind with biomolecule. So it means that when a student use these kind electrodes, and then so they need to polish it and to measuring their performance and characterize them and to see how is their structure of here is 111 and here's 100, and then they make sure to have a lot of 111, and then they will pursue following by the bioconjugation. So that's for only one test. But the problem, so as I mentioned in previously, the cost is $250 is not high for one electrode. But for the experiment wise, and we may use a lot of electrode because we need to gather a lot of data. And in general, each data may need, each figure may need 60 to 100 points. So if we polish the surface each time, and it's very time consuming and not efficient. So in previously, about 10 years ago, and we developing homemade electrode and use this tiny electrode, and we fabricate about 200 electrode and for the experiment. And we also developing our detection system. And recently, we found that they may have some commercial available prostate, and which is very convenient. So we change our system to commercial available prostate, and even use our homemade electrode, and we still can get some good result. At the beginning, we test the enterovirus as an example. Why we detect the enterovirus? Because first of all, the enterovirus it's an epidemic and invasive pathogen, and it may cause a lot of children being infected. For instance, in America, and there are more than 10,000 million children being infected, and it may also cause a lot of complication. And it's also outbreak at the East Asia or Southeast Asia, such like the Taiwan, Malaysia, China. So it's the very prominent virus if the patient being infected by the EV71. Because this virus is really prominent, and the patient may die with 1 or 2 days after admission, and they have a lot of neurologic complication, and in average is about 3 days, and their mortality rate, it can up to 26%. So in severe cases. So if we can detect that, and we know, and then we will know how to keep the patient from other patients, and then may reduce the infection to other patients, and also the patient can receive better care, and may reduce the mortality rate. Okay, so we use this system, and before that, here is the statistic data which provided by the Taiwan CDC, because we collaborate with them. And here is the data, and here, and you can find that the top 5 typings enterovirus from 2001 until 2024, and the EV71 in previously years and will be high. However, you can find that during the pandemic duration, and the EV71 become less. Why? Because every children has already been taking very good care, and prevent to interact with other children. But right now, this data is updated to April 1st. And there is 91 cases in Taiwan, and it's behind the Kozaki A6 will be the top one. Okay, so means that the typing is important. And why we need to detect that? Because EV71, their transmission can be infection patients in a very low titer. And if they are close contact and their latent period is 2 to 10. So if we have an ultra high sensitivity test, so we can detect rapidly, and then we can isolate those patients and prevent their further this kind of virus spread down. Okay, so let's see what's the typical protocol by CDC Taiwan. In general, when they receive the severe, when the hospital report some severe cases, and then those hospitals may take the sample from patient, and then those samples will be sent to the CDC and their contract laboratory for the virus diagnosis. First of all, they need to culture the virus and then to see whether they will have the cytopathic effect. And after that, they will transfer into the extract the RNA and transfer into the cDNA, or they can use the real-time PCR to detect. And another process is that they can use the RT-PCR, and currently WHO may use the cohort. And let's see the detailed process recommended by the WHO. And the RNA will be converted to the cDNA and amplified with the cohort primer and for the first PCR process, and then with the next PCR process to confirm whether they can be amplified or not, and then sequence and to determine what types of these enterovirus. And for the sequence duration is 1 to 2 days, but for the culture, for the cell culture and the process may take weeks. So if based on the rapid development of the EV71 cases, so it's not good for the patient. So that's why we start to collaborate with the CDC, and we try to detect the EV71 through our electrochemical biosensor. And first of all, we got the EV71 antibody from CDC, because based on the regulation, the EV71 antibody is not for the research purpose, and that can be only used in the hospital or the CDC. So we collaborate with them and got the antibody, and then those antibody can detect the virus. So we try to use the electrochemical process devices to detect, and it's a kind of antibody antigen process. So basically, and we can detect the EV71. And we found that the viral binding time can be saturated around 3 minutes incubation, and then their sensitivity is high, and their lowest detection is around 1 copy per reaction. It's indicated that we have developing the biosensor for EV71. But the problem, so compared to other current available EV71 detection method, and our platform is relatively good than others. So we use the similar system, and we can detect the human hemoglobin typing. Yeah, and you may know that for the haptoglobin and the each human may have the one type of haptoglobin, and there are three types haptoglobin. One is the 1-1, and another is 2-1 and the 2-2. So different people may have a different typing. So means that their resistance may be different. And then we have the antibody, so we can bind the haptoglobin and to detect their impedance change. So we can make the haptoglobin phenotyping sensing. So this indicate our system is ready. In my lab, and we developed this one, and we also can use some single domain antibody to detect some virus, some cancer cell, of course. But the problem or the bottleneck of the biosensing probe development is that, first of all, we need to have a pair of relator. Means that we need to get the antibody or some high affinity binding molecule. But if it's a new disease and it's outbreak rapidly, it's not easy to get more functional antibody or more specific antibody. So what can we do? And especially such like the EV for the enterovirus, and currently there is only EV71 antibody. However, if those virus, EV enterovirus, not EV71, and if they are variant, and we may not detect it. So that will become other problem. And the secondary problem is that the biosensing probes development is also a problem. As I mentioned, that each detection may take about 60 points or more. So for the conventional electrode, and the people may need to polish the electrode and then to make the detection. So it's a really time consuming, and especially if the manpower is there, it's require a lot of manpower to functionalize the surface. So what should we do if we need to screen rapidly? So we think that we try to use the thin-film electrode, developing a thin-film electrode by metal sputtering approach. In conventional, the growth of the metal sputtering on the surface, and they may have the different growth mechanism. However, as I mentioned, that if we want to conjugate the biomolecule with the metal surface, those nano structure and such a nano island or state will be better. And at the same time, since it's an electrode, so we need those islands and their substrate, and they may be conducting. So it's important to generate SK-like surface. And based on the requirement, means that this kind of SK model and may taking place around 1 or 2 layers of metal sputtering. So in previously study, they found that when the deposit of those metal granule on the substrate surface, and then they will lateral growth and to form a layer. But in fact, people found that such like this, if they sputtering like these, like nano islands on the surface, and put for a longer time, and then they will migrate and to form a plate. So it's a kind of the migration on the surface electrode. So it means that if we use the sputtering surface, and if we sputtering the metal ion granule on the substrate surface, and then although they may have some nano island or nano structure on the surface, but But eventually, and for the time being, and because the thermal migration and their conformation may change, and then not good for the bioconjugation. So what should we do? And we found out if we in previously study, people found that if they want to generate one stable 111 structure, and they need to use the silica surface with 111 as the substrate. So it may remain however, this kind of crystalline substrate is relatively expensive, not good for the biosensing purpose, especially if we need to polish the electrode again and again. It's not convenient for the point of care detect or on-site detection. So it's important for us to developing a surface active electrode. So in my lab, and we developing during that time, the palladium is relatively cheaper than gold. So we use the palladium to sputtering on the substrate. So the substrate choice is also important. And we found that the PET surface and their surface already with some microstructure because it's a polymer. So you can see these image, and they're roughly it seems like smooth. However, enlarged by the SEM, you can find that there's a lot of nano island on it, and their thickness is about 20 nanometer. By use the SRD indicate that, and those palladium structure is a P111 like conformation, and by analyze with the AFM also indicate that. And then here is the microstructure on surface structure of the PET. You can see that there's a lot of nanostructure on it, and so when the palladium deposit on it, and they may form the nanostructure on it, and then this kind combination, and they can keep the electrode stable and easy to be conjugate with the biomolecule, and their CV response is similar to the gold electrode. So we developing this kind of electrode, and this one will be used as a single-use electrode. So when we developing and we test that whether we can conjugate the biomolecule or not, so we use the DNA as a case, and we use a style modified DNA, and then we detect some specific drug, and we can find that some drug can interact with the specific sequence DNA, such like the some drug may recognize the SP1 sequence, and some but not recognize non-SP1 sequence. So it can be detected. And for the dosolubinsin, okay, no matter what kind of sequence, they can interact. So this indicates this kind of surface active electrode has been developed. Okay, so back to the COVID sensing. And since the virus variant is changed, the mutation is rapid. So it will be not important, not only sensing, since the typing or the neutralizing antibody will be the neutralized antibodies quantification is also required. So in 2020, and we try to developing this type biosensor to detect COVID-19. And here is the cases in Taiwan. And you may find that there's almost with less cases in Taiwan, that region. And our government also regulate regular researcher to assess those viruses. So how can we developing such biosensor? Luckily, just in April 2020, and the people already published from Wuhan, and they published their study indicate that the virus through its spike protein and interact with the human ACE2. So right now, we don't have the antibody, but we know there's interaction pair, and we know there are spike protein and ACE2, and we know they are sequence, and we also know their binding site. So we can recombinant express those proteins through the expression bacterial expression system, and then we got the protein. In general, people, but however, they are in the because we need a lot of functional protein. So we make them expressed in the inclusion body, and then we can refold it. Because my lab is expertise in the protein folding, so we can refold the protein through the overcritical reaction pathway. Means that we find that the protein folding the transition line may have a boundary. So we can use change the solvent environment of the protein, and we can refold the protein from the unfolding state to the native state continuous. So by using this way, and we can get the soluble protein. So we immobilize the ACE2 on the palladium electrode surface. And we can use the Raman spectrum to see how they binding. And we found that we only need about 800 nanogram sample, and those interactions can be detected. And we found that the AC2 bind with the palladium surface through the palladium style binding. And it's important that the only merely 2 granular layers surface is better than the very thin surface. So this kind of electrode, and we can detect the SARS-CoV-2. Okay, and since we make this kind of thin film electrode, and the volume for the detection is only 1 microliter. So, and the detection time is within 20 minutes. And even for the drug screening one, and that will mention that later, is about 4 microgram. Okay, for the EIS detection is within 1 minute. First of all, people may think that the refold protein, whether they are functional or not. And in our study, and it's quite, and first of all, as I mentioned, that we can immobilize the AC2 on the electrode surface, and then we test whether the spike protein bind with it or not. And here is the interactions ratio, and you can find that, and their dissociation constant can be determined. And this also indicate that both spike protein and ACE2 are functional. And we use this kind of probe, and we can detect some pseudovirus. And we also found that this pseudovirus can recognize this ACE2, and we can detect their dissociation constant too. And at the same time, during that time, and we can also detecting some mutants, European strain. And my collaborator, Professor Si Ru, and she thinks that since we have this kind of detection instrument, and whether we can detect whether those virus can store maybe still activate after the cold storage. So we make a test we found that after 1 month cold storage at -20 degrees, and those virus still can be detected. So what's this meaning? In 2021, the WHO delegation and China's investigation team worked together, try to figure out what's the origin of the SARS-CoV-2. And then they, after their investigation, and they got the four kind possible pathway for the virus infected. And the one is directly zoonotic induction. However, since it already break out for over years, and in fact, they cannot get any evidence. And they may also think that they have three indirect routes, such like the zoonotic infection from handling infection from animal, but they don't have the evidence. But they think that the most possible one is the zoonotic induction through the consumption of the contaminated food or the food from the infected animal. So they cite our work, and they say, okay, and the SARS-CoV-2 can remain infectious when frozen. Therefore, Ting felt it could not rule out this kind of possibility. Okay, so that's the only evidence. So since we have this kind of study, and we use the ACE2 and the spike, and we try to screen what kind of drug and we can use for may affect spike and ACE2 binding. And we found that the conformation of the ACE2 like a Pac-Man, and those drugs, the ACE inhibitor, and they may integrate into the cave of the ACE1. But there is no report whether the ACE2 will also be affected by this ACEI. So before we study, and there's a lot of clinical statistic study indicate that people may have the risk when they use those drugs. And this being cited by the scientific society. However, those prospection has been rejected by the major cardiologist scientific association, such like ACC, HFSA, and others, because they say, there is no significant evidence to support this kind of conclusion. So the patient may be safely. But no matter how, we use the molecular modeling, also collaborate with our collaborator in the University of Malaya, and we screen out 8 potential lead. And we found that they may have a bulky group ring at the amino terminal of ACI. So we use our system and measuring their interactions and become a spike protein and ACE2, and we found that 4 of them, such like the peridopia or ramapil, and they may reduce the spike protein binding, and from 48% to 42%, or from 42% to 72%. However, another 3 ACEI, and they can increase the binding affinity, and from 25-fold to even to the 170-fold. And they are the enalapiel or lisinapiel. So the similar trend can be found in a real virus detection. And so this indicates that this study can be used to detection the protein-protein interaction, and may also study in the anti-infection drug screening. So, and other teams, researchers, and they take our detections approaches, and they can detect some doping-based receptor and thrombotic markers interaction, and which published in the Nature Chemical Biologist. So this indicates that this kind of study is powerful to study the protein-protein interaction or other drugs interaction too. Okay, since we as I mentioned previously, the EV enterovirus disease is sensing is important, and their typing is also important. So we can detect the different typing of the EV71, and this data is in the revision and will be published soon. And people found that even the EV71 and they may highly sequence homology to the Kawasaki 16. And so how Kawasaki A6. So how can we distinguish them? So luckily we have the sequence, and then we can design some specific probe and to detection layer binding. And here is a result. You can find out that we can tell the EV71 and with other virus sequence. So we can type in them. So in summary, right now we're developing a biosensor platform for identify the SARS-CoV-2 and its variant. And this can also be used for the drug screening, and even the enterovirus can be typed. And this platform can be adapted for identify drug against other types of infectious disease or some novel infectious disease too. Okay. So this kind of work, and with a lot of support, not only from the NST National Science Technology Council, and also from the Ministry of Education Taiwan, and also with the team supported by the National Yangming Jiao Tong University, Chang University, and Chang Hospital, and the National Cheng Kung University, and the Taipei Veteran Hospital, and the University of Malaysia, and the CDC Taiwan. And here is the team of us, and I think that it's important to combine both strengths together, and then we can get the most powerful and useful tool to solve the problem of the new infectious disease outbreak. So here is the contact information, and you are welcome to contact me. And thanks for your attention. Thank you very much.
All right, thank you very much, Prof. Chang, for the very insightful presentation. And we can clearly see the impact of your work has been used for the WHO report. So it's an outstanding achievement. Right, I would like to invite the audience who would like to ask directly to Prof. Chang about his presentation just now.
Okay, I'm Nurul Huda from Universiti Kebangsaan Malaysia, and the same group with Dr. Izzati. Just out of curiosity, you're using palladium nano film, right? Have you used other metal? Does it shows a similar effect as well, or similar efficiency as well? Or any reason why palladium?
Yeah.
Okay.
Oh,
If I can iterate back the question from Dr.
Yeah.
Nurul Huda.
yeah. Sorry.
You are using a palladium-based nano-thin film, right, for the construction of your biosensor.
Yeah.
Have you tried with other metals from palladium?
In fact, not only the palladium, and right now, because currently the palladium is almost the price to the gold.
Yeah.
So that's why we already use the gold to developing the new film, and we already done that. Okay, yeah, because in fact the palladium is when we use that, and you may think that 7 years ago the price of the palladium is the 2/3 of the gold, so that's why we use that. And currently we have already found that we only need merely about 20 nanometer synchronous metal layer to prepare those electrodes. So either gold or palladium will be good for our study. So currently, my industrial collaborator, and they will use the gold as the target to prepare those electrons. So currently we are doing that. In fact, there are also other commercial available electrode which sold in a disposable electrode which sold in by other company, but their price is not friendly. Yeah.
Prof. Chang, if we can see today that there's a lot of application on this nano-thin film, especially in this infectious disease detection. But of course, before we can get this technology to the clinical setting, what kind of challenge that you foresee? I mean, in making this NTF, this nano-thin film, into a large-scale implementation for clinical setting, main challenge that you can foresee?
I think that it will be still the same problem. Okay, so let me show you. We still face the problem is that how could we, first of all, for the application part, and it's important for us to get the proper interacting pair. Yeah, especially for those infectious disease. And if we want to use to detect their surface protein, and then we need to get their interaction pair. But if they are novel infectious pathogens, and it's not easy to know what kind of protein they are or what kind of sequence they are. However, their sequence is quite easy to be get. So that's why we try to use the typing approach, and it means that we can detecting some specific sequence of the DNA sequence or RNA sequence. So by using these kind of approaches, and that will be faster to detect this kind of structure. Okay, and another issue is that for the this kind of metal sputtering, and as I mentioned, that regular sputtering method and those metal ion and metal granule, they sputtering on the surface, and then they may form the active structure. However, if you put for a long while, and due to the thermal migration, and their conformation may change. So their surface activity may reduce. But if we can use some chemical modification combining with the approaches and then retain the structure of the surface, or we can make the further application more systematic or robust. That's what we're trying hard and to develop this.
Right, let's go in with it. Any more questions from the participant today? We also stream live on the faculty's Facebook, so I'm going to check very quickly if we have any question from the Facebook. Okay, I have one question.
Okay.
Okay, it's from Lou. Is
Lou.
EIS a user-friendly method for point of care application? I think POC is point of care, right? Application.
Sure.
Yes,
And in fact, for the point of care, that's the purpose what we would like to pursue. So it will be friendly method for the point of care and even people. So that's why we try to developing some disposable electrode for even people can use that easily, such like the blood sugar test. So that will be useful. And the problem is that if we want to use for point of care, first of all, we need to get the permission from the Ministry of Health Department and got a certificate. And then, because this thing is a kind of medical devices, not for research only. So that will be in the future. So that's why we need to collaborate with not only the hospital, and we also need the industrial partner and to join us and to get the approval by the bio device approval. Yeah.
Yes. Yeah, because the electrode has to come to the industrial scales
Of
and we have
course.
to get
Yeah.
a bio devices. And it's not easy. Okay.
Yeah, but in fact, its operation is quite easy.
All right. I think if we don't have any more question, that will bring us to the end of our question and answer session. A very big thank you to you, Prof. Chang, for sharing your expertise with us today. We hope in future we will more collaboration with your university, with the group, with our biosensor and chemical sensor group in UKM. So thank you again for joining us and have a great day today. And we hope to see you in our next webinar. All right. Thank you again, Prof. Chang. See you,
Thank you.
everyone. All right.
Bye.
Bye-bye.